back, students. Hope you are doing well. As you know that as promised before, again we are back with a short series of biology concepts. So very good morning, good evening. जब भी आप जिस टाइम पे इस वीडियो को देख रहे हैं, have a good time ahead. मैं चांदनी शर्मा आपकी बायोलॉजी मेंटर एक बार फिर आपके सामने हाजिर हूँ कुछ हॉट टॉपिक्स लेके जो आपके नीट 2026 में आपको बहुत काम आएंगे, राइट? तो आपको पहले भी बोला था कि ये हमारी एक शॉर्ट रिवीजन सीरीज है जिसको हम आपके हाफ ईयरली एग्जाम्स को मद्देनजर रखते हुए हमने इस चीज़ को डिज़ाइन किया है। तो कुछ इंपॉर्टेंट टॉपिक्स जो आपकी एग्जाम में डेफिनेटली आपको काम आएंगे, उन्हीं टॉपिक्स को हम लेके आज एक बार फिर आपके सामने हाजिर है इस नई सीरीज के साथ। एज आप सब लोग जानते हैं कि ईजी बहुत जल्द अपने एक ऑनलाइन कोचिंग प्लेटफॉर्म को लॉन्च करने वाला है, दैट इज ऑन 1st of October 2025। तो fasten your seat belts and get ready for the new drive with the EG classes। इसके डिटेल्स आपको नीचे डिस्क्रिप्शन बॉक्स में मिल जाएंगे। अगर डिटेल्स आपको और चाहिए तो एक फ़ोन नंबर ब्लिंक कर रहा होगा, आप उस फ़ोन नंबर पे कॉल करके और ज़्यादा डिटेल में जानने के लिए आप हमें संपर्क कर सकते हैं। और इसके अलावा आपके मन में कोई जहन में कोई ऐसा टॉपिक है बायोलॉजी का जो आपसे मुझसे समझना चाहते हैं कि आप चाहते हैं कि मैं उस पर्टिकुलर टॉपिक की कोई वीडियो शूट करूं आपके लिए, उस चीज़ को एक्सप्लेन करूं, उस चीज़ के PYQs लेके आपके सामने आऊं, तो आप कमेंट बॉक्स में उस चीज़ को डेफिनेटली वहाँ पे आप लिख सकते हैं। उस चीज़ पे हम गौर करेंगे और बहुत ही जल्द उन वीडियोज़ को आपके सामने लेके एक बार फिर हाजिर हो जाएंगे। चलिए आज के हमारे इस सफर को शुरू करते एक नए टॉपिक से, दैट इज योर PGR ऑक्सिन, राइट? सो ये आपका फिर से एक प्लांट फिजियोलॉजी का नया टॉपिक है, दैट इज PGR मीन्स प्लांट ग्रोथ रेगुलेटर्स, राइट? सो इफ आई राइट व्हाट डू आई मीन बाय PGR, एक बार थोड़ा सा कलर चेंज, राइट? सो हमारे पास क्या है, PGR आज का टॉपिक है और उसमें हमने क्या लिखा है, ऑक्सिन, राइट? विथ रिस्पेक्ट टू ऑक्सिन आज हम सिर्फ और सिर्फ डिस्कवरी ऑफ ऑक्सिन की बात करेंगे। आपको अगर ये वीडियो अच्छा लगता है, आपको कंटेंट अच्छा लगता है, तो आप इस चीज़ को लाइक करें, इस पर्टिकुलर चैनल को सब्सक्राइब करें। आप कमेंट में लिखें कि आप और प्लांट हार्मोन्स के इस वीडियोस को जानना चाहते हैं और प्लांट हार्मोन्स के बारे में हमसे पढ़ना चाहते हैं, तो हम अगली सीरीज को डेफिनेटली दूसरे हार्मोन्स के साथ लेके आएंगे आपके सामने, राइट? तो आइए, व्हाट डू आई मीन बाय PGR? PGR is an abbreviated form of plant growth regulators, right? So word मैंने यहाँ पे क्या use किया है, it is an abbreviated form of plant, G stands for growth and R stands for regulators, right? So those chemicals, right? Please mind my words, मैंने क्या लिखा है, those chemicals which are responsible or which regulate the plant growth, they are known as what? Plant growth regulators, right? So you might be familiar about the hormones which are secreted in the human body, right? They are also known as animal growth regulators. Same way, plants also have certain chemicals which are secreted by them and they are used in the growth of they even promote the growth and they inhibit the growth, and some of the hormones will have dual function. In a short while we'll make a mind map of that as well, right? So what are these are plant growth regulators. So if I write their another name, they are known as what? They are known as phytohormones, right? So if I write the word phytohormones, phyto has to do with plants and hormones are the certain chemicals which are secreted by living organisms. So phytohormones is very specific related to those chemicals which are secreted by plants are known as what? Phytohormones, right? So what is this PGR? PGR nothing but it is your plant hormones, phytohormones or plant growth regulators. These are the synonym words for the same, that is plant hormone, right? Okay, now if I talk about these plant hormones, then these let us write down few of the characteristics of these plant hormones, right? So what are the characteristics of PGR? If I talk about characteristics of PGR and if I write the first characteristics of PGR, then what it will be? So first, it is nothing but it is a chemical substance, right? We are talking in general about all plant growth regulators, right? So we are not writing that what are the derivatives of the respective plant growth regulators, but in general they are chemicals which are secreted by plants. So they are chemical by nature, or they are chemical substances, right? And of course they are hormones, so they are secreted in trace amount, right? If I say trace amount, means I can write trace amount, or I can write they are secreted in small quantity, means they are not secreted in higher concentration, but they are secreted in very less concentration. We know that in humans also the concentration of hormones like adrenaline, right, pituitary stimulating hormone, or you can say insulin, not pituitary stimulating hormone, I'm sorry for that word, but one that is adrenaline, then another hormone you have that is insulin, right? They all are human, you can say hormones which are secreted by humans, right? So they are secreted in minute quantity, or in trace amount, or in small quantity, right? So the second important characteristics of this PGR is that it is secreted in trace amount, or it is secreted in small quantity, right? Now what will be the third characteristics? Maybe the location of the hormone, right? We have studied that certain hormones, they get stimulated from our pituitary gland, and their mode of action is somewhere else in the body, right? So the same way here, they may be secreted at the location, or they may be secreted away from the location, right? So the third characteristics, if I write about its secretion, then secretion can be at the location of action, mean where the action has to take place. At the location of action means to which particular organ they will be stimulating, or it may be away from the location, right? So it may be where, it may be away from the location. So these are known as what? Such type of chemicals are known as what? Plant growth regulators, right? So hope I am clear with what are the characteristics, or if I have to define a plant growth regulator, then what it is? It is a chemical substance. Second important thing, it is secreted in trace amount, or in small quantity, or in micro quantity, we can say. These are the synonym words. And third, they may be secreted at the site of action, or they may be secreted at or away from the site of the action. Like if I give an example of this to elaborate it, then if you remember, we had done in sexual reproduction in plants, these were what? Your shoot apical meristem. From the nodal region, what was formed? A leaf was formed, right? Now under the influence of a hormone, that is what? Florigen. Under the influence of the hormone florigen, which will travel from the leaf and it will go to shoot apical meristem, and it will give rise to what? Floral primordium, right? From floral primordium, what will arise? A flower will arise, right? So if I say for this florigen, a group of hormone which is responsible for inducing flowering in the plant was secreted from leaf, and it traveled to the shoot apical meristem, SAM, and there some of the shoot apical meristem were converted into floral meristem, and floral meristem indeed produce a floral primordium, and floral primordium will give rise to floral bud followed by a flower, right? So they may be secreted at the location of action, or maybe away from the location of action. Such group of chemicals are known as what? They are known as plant growth regulators, right? Now if I talk about the types of plant growth regulators, then your PGRs, that is plant growth regulators, can be divided into three basic categories, right? One which is going to regulate the plant growth, right? So they are going to regulate plant growth, means they will help in the growth of the plant, right? The second, they may inhibit the plant growth, right? So if I write here inhibit, so I'll write a precise word here, that is they will inhibit, first we'll write down this and then complete this. They will inhibit plant growth. If something is inhibited, then here something has to be promoted, right? So they will promote plant growth, they will inhibit plant growth, or certain plant growth regulators, they are dual by nature. Means they are going to act as promoter as well as they are going to act as inhibitor. So such type of plant growth are known as what? They are known as dual by nature. So they are promoter as well as inhibitor, right? So what would be the characteristics of this plant growth promoters, or those plant growth regulators which are going to promote the plant growth? So how they will promote the plant growth? They will help in what? Of course, growth means cell division. So they will help in cell division. They will help in the process of elongation. You can say plant elongates and they will help in the process of elongation, cell division, right? And inhibitor means they will carry out the process of aging, right? Means budhapa, buddha hona, senescence, aging, right? Followed by, they may result, if they are secreted, they may result into dormancy, right? So when the conditions are not favorable, the plant will not grow, it will remain inactive for a period of time, so your plant growth is inhibited. So the word we use is, that is what? Dormancy, right? So what are the characteristics of inhibitor, plant growth inhibitors? They will induce what? Aging. Aging is also known by another name, that is senescence, followed by dormancy. That means the plant will remain dormant for a period of time, right? And what promoters will do? They will help in cell division, they will help in cell elongation, right? In this way, they will help in plant growth, and here they will cease the plant growth, or they will stop the plant growth, right? So, and dual nature means certain hormones like ethylene, they will act as both, they will promote as well as they will inhibit the growth. That we will study in detail when we will talk about ethylene, right? Now today we are going to focus on one plant growth regulator, that is what? Auxin. And auxin acts as what? Auxin acts as your plant growth promoter, right? So one of the plant growth promoter whose discovery we are going to study, that is what? Auxin, right? So what is auxin? If I say auxin is a phytohormone which is responsible or which promotes the plant growth, right? Okay, fine. So now let us start with your PGR, that is your auxin, right? So if I write with a bold handwriting, the first phytohormone what we are going to start today, that is what? That is your auxin. So my first question to you is, auxin is inhibitor or promoter? Of course, it is what? It is a growth promoter, right? So henceforth, whenever we'll be talking about the promoter part, that is how it will promote. So everywhere we'll be talking about cell growth, cell division, cell elongation. Nowhere we'll be talking about ceasing of the plant growth or plant growth is not taking place. Am I clear with this? Okay, fine. Now let's start with first important thing, that is the discovery of auxin. Right, so the first topic what we are going to do, or the topic, what sole topic we are going to do in the today's video is discovery. Right, so whenever I talk about the discovery of auxin, the first person or the biologist which comes in my mind is Charles Darwin, right? The name Charles Darwin, we are very familiar with this name in the field of biology. He is known as the father of biology. The same Charles Darwin showed his contribution even in the field of plant physiology indirectly, right? So Charles Darwin, along with his son Francis Darwin, right? So who are the two scientists who discovered, who started with the, or who were the pioneers of the discovery of auxin? You can say Charles Darwin and Francis Darwin, right? So what they were doing, if I talk about, we'll write down the name, that is C. Darwin, that is Charles Darwin, and his son, you can say Francis Darwin, right? They both discovered a chemical substance, right? So what was their discovery? They discovered a chemical substance. How did they discover this particular chemical substance? It was Charles Darwin when he was studying the concept of phototropism, right? What he was studying the concept of phototropism. We know that what is phototropism? Plants, they show tropic movement, and the movement of plant towards the sunlight is known as what? Phototropism. If you wish to know about in detail about phototropism, you can drop a comment on the link box that you want to study in detail about the tropic movements of the plant. So we'll come up with the next video which shows that tropic movement of plants, right? So Charles Darwin was studying what? He was studying the process of photoperiodism, right, in plants, right? Now, in which plant he was studying this particular phototropism? Then he was studying in the plant that is oats, right? Of course, it is the same safola oats जो हमारे kitchen में मिलता है to make your figure very fit and fine, right? So the same oats plant was used. So what he did, what he was actually studying, he took a well potted plant. So this is a pot, right? And if I draw, this is a plant, right? So these are the leaves, and down you have a root system, right? So this is the root system. So he took a well potted plant, and यहाँ पे उन्होंने क्या डाला? A source of light, that was sun, because he wanted to study what? He wanted to study photoperiodism. So he found that, else it was dark enough, right? He found that the shoot started moving towards the light, right? So what he observed that this particular plant, it started showing the movement towards the sunlight, right? So he called this movement as what? Phototropism. So this movement of plant towards the light made him curious that what was responsible for this particular thing, right? There would be something in the plant because of which the plant showed the movement towards the light. So that curiosity of Charles Darwin landed up with the discovery of Auxin, right? So what he did, that in the next slide we will study, he took the shoot tip, and in shoot tip we know that we have a structure that is known as what? Coleoptile, right? So in a well potted plant, from this he took what? Coleoptile, and he cut this coleoptile, right? So the same oats plant he took, right? And he first, let us understand like this. If I take this well potted plant, this is my coleoptile region. We know that the embryo or the seed contains embryo has two main parts, that is radicle and plumule, right? Then we have hypocotyle, epicotyle, then we have coleoptile, coleorhiza, right? So the प्लिम्यूल एक ऊपर टोपी पहनता है इन केस ऑफ मोनोकॉट, जिसको हम क्या बोलते हैं? कोलियोप्टाइल, राइट? द सेम कोलियोप्टाइल इज फाउंड हियर. सो वी आर टॉकिंग अबाउट द सेम कोलियोप्टाइल which is found in the shoot system of a plant, right? So when this particular plant was exposed to what? When he exposed this particular plant to, if I don't draw sunlight, let's just draw a bulb, right, to make it more interesting, right? So this is your bulb, right? So from here the lights were falling on this particular coleoptile, right? And what he observed after a period of time, that this coleoptile, it showed the bending towards the light, right? The only light source which was available nearby, right? So here was the light source, and he observed that the bending took place on the right side, right? So here he got curious that why this bending was shown by the plant. So what he did next, that is, he removed the tip or the coleoptile of the stem, right? So what he got afterwards here, he will have only the lower portion of the shoot, right? And then again he exposed this lower portion of this shoot to what? To a bulb. If I draw like this, it will be more clear, a filament of a bulb, right? So here what was observed here? No bending was observed, right? So what was the conclusion? So there was no bending. So there was something in what? In this particular area, that is coleoptile, which was responsible for the bending. So what was the conclusion of the Darwin? That there is some chemical substance, right? A chemical substance which is found in the coleoptile, which is responsible for the bending of the shoot tip, right? So he had no idea what this particular chemical substance is, right? So now we are ended up with what question? We have now a new question in our mind, that what is this chemical substance, right? So who will give the answer of this question? Of course, some another scientist. That it was in the year 1913, right? So it was in the year which? It was in the year 1913, right? A scientist named Jensen, right? A scientist whose name was B. Jensen. He came up that, okay, I will find out that what is this particular chemical substance, right? So what he did was, he again took the same plant, the plant which was taken by B. Jensen was which? It was an oats plant, right? So he took an oat plant. Same way he took what? He took a plant whose coleoptile was present, right? So this is an oat plant. And what is this area? This area is coleoptile, right? So what is this particular area? This area is what? It is the coleoptile area, right? Now, by the time they knew that there is something present here, right, which is responsible for the bending, right? So what he did was, he cut this particular, what he did, he cut this particular coleoptile piece, right? So now he was here with what? He had a shoot tip with him, and this shoot tip is what? It is your coleoptile, right? And interestingly, what he did, he put this particular shoot tip on what? On an agar block, right? So what is this? This is your agar block, the same agar powder what we have studied in what? We have studied in, yes, the plant kingdom, right? Now what he presumed, that as it was a chemical substance, that means it may be diffusible by nature, right? So with this presumption in his mind, what he was trying to do, that from here he presumed that this particular chemical will start diffusing in the agar block, right? So from here I'll just select this, clone, keep it here, right? And his next observation was what? Let us erase this arrow. This is coleoptile, right? From here, this particular chemical will diffuse where? It will diffuse in the agar block, right? So chemical substance which was present on the tip of the shoot, right, which was responsible for what? Which was responsible for the bending, right, is now being diffused in the agar block, right? Now what he did is, next he a series of experiments were done to discover this particular, you can say, chemical substance. So here what was done was, a cut coleoptile was, a cut shoot tip was taken, means there was no coleoptile present on the top, right? So this is your what? A shoot. Now this particular agar powder, agar block, which showed the presence of what? Diffused chemical, right? So in this agar block, what was there? The chemical which was present in the coleoptile has diffused in this agar block. So one was taken, the agar block with the diffused chemical, right? And this you can write down as what? As A. And in the B portion, they again took a shoot without coleoptile, right? This is a shoot without coleoptile, and an agar block which will act as a control. Means inside this what was not present? Inside this, the a diffused chemical was not present. Now when these two chemicals were exposed to sunlight, so if this is my bulb and they were exposed to what? Light, or you can say source of unilateral, right? From one direction the light is coming, right? So in this way, if this is a bulb and they are exposed to what? Light, right? What did they observe at the end? That there was no growth observed in control, right? What did they observe? No growth in the control, and the growth was observed on the shoot which had agar agar with that particular diffused chemical, right? So what was the conclusion? The first conclusion that was made, that the chemical substance about what the Darwin was talking, right? The chemical substance about which the Darwin was talking, the first important characteristic is what? It is diffusible by nature, right? Means it can be diffused from one medium to another medium, right? So this is about the first important characteristics of the chemical substance, which was on the way of the discovery, right? What was that? This slide shows that the chemical substance is what? Diffusible. From the tip of the coleoptile, it got diffused where? It got diffused in the agar block, right? And from agar block, when it was kept here, it was diffused. Maybe the cells of the you can say shoot region absorbed that chemical, and they started growing what? And they started showing what? Growth, right? Now the next question is the bulb which has been placed over here, right? So every time whenever the bulb or whenever the light was exposed, the growth was shown, and when the light was not exposed, the growth was not shown. So the second question which came into our mind was that, is there any relationship between the auxin and the light? Or there is no relationship between auxin and the light? If yes, then maybe the light, if the light is a, because light is always present, right? So what is the connection between the light and the auxin? So let us now study the second portion, that what is the connection between the light and the auxin. So now in the next experiment, what was done? They took a particular agar block, right? Again, what was taken? An agar block was taken, and they I think black color is not visible, right? And they, what did they do? They inserted a impermeable thing inside this agar block, right? So what was inserted? What is this blue color structure? What is this blue color structure? It is an it is an impermeable thing, right? Means it won't allow the entry of anything on this side. Entry is not allowed, right? What it can be? It can be anything which is made up of metal, right? So let us talk about, he took a blade, right? So he took a blade and he inserted into the agar block, right? After inserting into the agar block, this agar block was again kept where? It was kept on the, shall we make it small? Okay, fine, right? And let us move the selection a bit here. Let us shift this also a bit here, right? So now the things which are on the right side will remain on right, which are on the left will remain on left, right? And this was again placed on what? It was again placed on the shoot. This is what? Your shoot, right? So presuming that the chemical substance will move there, right? And here it was exposed to the light, right? So when it was exposed to the light here, the substance would have diffused into the agar block. Let us not draw right now that it has diffused into the agar block, right? This is your what? Agar block. Now this agar block was taken and it was divided into two parts. Let us make this one with this color and on this side with the pink color, right? So we have one block which is going to have what? It is going to have This particular shade means the one which is present on my left hand side, right? And the another block which is there, which is present on my right hand side, right? This is my right hand side, right? These two blocks were taken, and these two blocks were tied again to what? These two blocks were taken from here and tied to the shoot, right? The shoot which was present, they tied this particular to the on the left hand side of the shoot, and this was placed on the right hand side of the shoot, right? Again, both of them were exposed to what? Exposed to what? Light. This is what your light source, right? Let us clear these things first. They were exposed to what? They were exposed to the light source, right? And what was amazingly observed, that this particular block, it showed the bending, and here no bending or almost very less bending was observed. That means the mode of action of auxin, right? This particular auxin was present over here, or the chemical substance was present over here, is away from the sunlight, right? So what is the second conclusion made? Is there any connection between auxin and the light? Yes, the auxin is secreted in the area which is away from the light, which is away from the light, right? Or I can say in the shade area the auxin is secreted, right? So this was your light exposed area, this was not exposed area. So the mode of action is what? In the shade area. So here the auxin will increase the length of the cell, and ultimately the growth will take place, and this uneven growth, because only this side is growing, this particular side is showing no growth. So when this side is showing, it is slowly showing what? It is showing your bending. So thus we observe what? Bending of the shoot tip towards the light, because the cells away from the light are showing what? Are showing growth, right? So finally we came to a conclusion that this particular hormone which is responsible for the growth is known as what? Is known as auxin, right? So this is how the discovery of the auxin was made by a group of scientists, from where the pioneer was Charles Darwin and his son, that is Francis Darwin. Hope you liked the video. If you like the video, please comment, please subscribe, please like the channel, and do comment what other biology concepts you would like to learn from your biology mentor Chandni Sharma. And please don't forget that we are going to start, EG is going to start the online batches, a full course for your class 11, for your class 12, for your NEET from 1st of October 2025. So please do not forget to inquire on the phone number which is present in the description box, right? So all the best for your exams. Do well, study, give importance to all the subjects, and good night or goodbye. Thank you so much.